You are currently the only person in this conference.
I had a little crisis yesterday when I was going through your exam because I didn't realize I did not post a blended lecture that I thought was posted over CNS versus PNS. We went over it in lecture. We did the CNS versus PNS. We did the whole outline of the nervous system. We did all the cranial nerves, but I just got it on and I posted it. So that is optional. And it's the one, let me turn this on here. It's just kind of a good overview. And if you weren't here for class, to draw it out. So if you go into, and again, I thought it was posted, but it wasn't. All of the information is, okay. Introduction to the nervous system. If you read the PowerPoint on introduction to the nervous system, as well as the lecture notes, you'll notice that on this neurons functions recorded, it doesn't cover everything. There was actually a part before that that was CNS versus PNS. We did that in lecture, but I went ahead and posted this as well. So if you wanted to go back and just see the big picture and redraw it, I did that in as a blended lecture as well. So that's kind of a bonus. If you don't do it, it's fine. You'll still get the 1 point for it. I'll just go through and give everybody 1 point for it because we did it in class, but I wanted to put it up there in case you wanted to see it. The neuroglia and coverings was another activity, not worth points, but that's information that's on the test as well. So CNS versus PNS should be reinforcing, again, between what you've already gone over. A lot of this is reinforcing or going into slightly more detail than what we went over in with Dr. Kingsley. All right. I think I've got a PowerPoint here. I just did the review for motor for PHYS 2 because they're taking their test tomorrow. PHYS 1, exam 2, review. Let's see what that one looks like. Oh, it's a big one. This has been posted and available for you for a little while, so I'm assuming you've worked through it to some degree. Introduction to the nervous system and autonomic nervous system, CNS versus PNS. Everybody go with CNS versus PNS. How many pairs of cervical spinal nerves do we have? 8 pairs. How many pairs? 12 pairs of cranial nerves, 8, 12, 5, 5, and 1 to 3 sacral in the human. So you might get 8 pairs of nerves on the exam, but again, should be one of your easier questions. CNS versus PNS, you should know what belongs in the CNS, brain, brainstem, spinal cord. What belongs in the CNS, any branch of any cranial nerve or any branch of any peripheral nerve. So I gave you musculocutaneous nerve. Well, that's PNS. Again, fairly straightforward on that one. Questions might look something like this: which is a component of the peripheral nervous system, or all of the following belong in the central nervous system except. Notice how that's the same question, just worded 2 different ways. So you could get this version or you could get this version. In any event, it's a cranial nerve or any branch of a cranial nerve, a spinal nerve or any branch of a spinal nerve. CNS, what provides myelin for tracts? Tracts are in the CNS. Myelin in the CNS comes from what? Oligo, dendrocytes. Myelin for nerves in the PNS comes from what? Schwann cells. Supported nutrition for neurons that live in nuclei. Well, nuclei are in the CNS, so therefore this would be astrocytes. Support and nutrition for neurons that reside in ganglia. By the way, the ganglia and neurons for the peripheral nervous system arise from neural crest cells. You haven't gotten this in, and then the ones from the CNS arise from neural tube cells. Not going to test you on that one yet, but just starting to put everything together for you. So when you get into what makes cerebrospinal fluid? Ependymal cells. or ependymal cells. Either one works. Which ones provide phagocytic or immune function in the CNS? Microglia. Microglia. Okay. How many pairs of cranial nerves? 12. How many pairs of cervical nerves? 8. Thoracic. 12. Lumbar. Sacral. Coccygeal. By the way, this is in a typical human. There will be situations where I've got an extra thoracic. Okay. A lot of times, if you have an extra lumbar, you have one less sacral. If you have an extra sacral, you've got one less lumbar. It's called sacralization or lumbarization. All right, the central nervous system, PIA, is going to give us endoneurium. Make sure you watch this end as well because endoneurium is not endomysium. Arachnoid is going to give us peri, and dura is going to give us epi. If you get endo and epi confused, I have a song for you. Endo, peri, epi. It's not even a song, it's just 3 notes. Do it with me. Endo, peri, epi. All right, so epineurium is the big covering. What's that? No, just endo, peri, epi. You'll be like, oh, he found that question. No, if you can put a song, we got a lot more songs and dances that come in PHYS 2. PHYS 3 is boring. We don't get to sing and dance as much in PHYS 3, but PHYS 2 gets a lot of songs and dances. So we'll have fun. So watch out for the suffix. Remember neurium, metrium, mycium, cardium. So if I switch this out, watch out for the suffix on some of these. That's a medical terminology. All right, neuron cell bodies in ganglia, Schwann cells. I'm going to try to move along here because we already did this. What CNS structure becomes the perineurium? Endoperi is arachnoid matter. Yes. Oh, because I read the question wrong. You are correct. It is satellite cells. I read the question as myelin. You have to answer the question that's written, not the one that you think was written. This is what happens when you read a test question too quickly, too. I read that as what provides, and my brain put myelin in there, and I was like, oh, Schwann cells, myelin, moving on. No, this is what happens when we go too quickly. You are correct. This is satellite cells. If this said myelin, then it would be Schwann cells, but it doesn't say myelin. So you have to answer the question that is written, not the question that you wanted to be written. Sorry about that. That one is arachnoid matter, though. All the following are examples of somatic sensation. Okay, now this is somatic as opposed to what are our 2 other options for sensory function? Sensory function, we have 3 options for sensory function. Somatic means what? Body. So somatic means body, and we have within our somatic sensations versus what? Visceral versus what? Special. So we have 3 categories of sensory, and each of these should fall into either somatic, visceral, or special. All right, touch, what is it? Touch is somatic. Smell, special. Pain, somatic. Proprioception, somatic. And temperature, somatic. Although pain can also be visceral. You can have visceral pain. It's a different pattern. So in this case, it's going to, but all of these are still somatic sensations as opposed to special sensations. What are other special sensations? Seeing, hearing, smelling, tasting, balance. Touch is the only one that's not. What's the conversion of environmental stimuli into neurological electrical impulses? That's a definition question. What was that definition of? Transduction. Transduction is the term for the conversion of environmental stimuli into neurological electrical impulses. Where does this happen? At peripheral receptors of pseudounipolar or bipolar neurons. So transduction occurs at the peripheral receptors of pseudounipolar neurons. It's where we're going to have tactile receptors. nociceptors, mechanoreceptors, thermoreceptors. Does that make sense? So this goes along with our somatic sensations. Peripheral receptors of pseudounipolar neurons is the location where transduction of environmental stimuli into neurological impulses will occur. Where does the action potential originate as the electrical current is coming in from the receptor? Where does the action potential originate on a pseudounipolar neuron? First node of Ranvier. All right, good information. Which is classified as a special sensation? Taste. Which are visceral sensations, such as baroreceptors, chemoreception, stretch. Those would be predominantly, although stretch is also a mechanoreception. But baroreceptors are for blood pressure. Chemoreceptors are for the concentration of substances in the body. So taste is a special sensation. Touch, proprioception, and temperature are all somatic sensations. And then visceral sensations, these are a couple of examples of visceral sensation. Motor and integrative function, which is a somatic motor function. Okay, somatic means what? Body, and in this case, it means voluntary. So our motor, if you see somatic for, it's going to be voluntary, and voluntary tells you what? Skeletal muscle. Voluntary innervates skeletal muscle. So when you see somatic, you say skeletal muscle. All right, so now we go somatic equals skeletal muscle, and voluntary. Increase in heart rate, what is that? Autonomic nervous system. Secretion of sweat, what is that? Autonomic nervous system. Contraction of the pyloric sphincter, I don't know where that is, but it's probably the autonomic nervous system. Contraction of the pupil dilator muscle. Autonomic nervous system. Contraction of the biceps brachii. Secretion of saliva. So that's just a voluntary versus involuntary. Somatic motor function versus a visceral motor function as our options. Integrative functions of the nervous system usually utilize interneurons of the central nervous system. Where'd you go? Which is under voluntary control, well, if it's a sense, it's probably not under voluntary control, huh? What's is under voluntary control? Special senses? Nope. Skeletal muscle? Yep. Cardiac muscle? Nope. Smooth muscle? Nope. I don't know why the computer auto-generated that one. That should be up there. Whatever. Okay. Most afferent neurons are made up of axons. Most afferent nerves are made up of axons of what type of neurons? Most. What does that mean? Pseudounipolar. Which cranial nerves utilize bipolar neurons in their pathways? Cranial nerve 1, olfactory. Cranial nerve 2, optic. Cranial nerve 8, vestibular and cochlear are going to utilize bipolar neurons in their pathways. This is a short list. It needs to be somewhere in your memory bank. That's your bipolar neuron list. Which activates alpha or beta adrenergic receptors on smooth muscle, cardiac muscle? And I took this question out because I said we don't need to know neurotransmitters yet. That's for the next test. Also, glutamate and GABA came out also on the next test, although I think you knew this one. What do we know? Almost always excitatory in the brain and brainstem. Glutamate, almost always inhibitory in the brain and brainstem. GABA. And in the spinal cord, GABA sister, glycine. So yeah, you would have known that one, but because I didn't put it on the PowerPoint, etc., it might show up on the next test. Which cranial nerves use bipolar neurons? 1, 2, and 8. And notice how 2 and 8 are also your super special senses. Those are your SSA. one is a SVA. All right, which contains the nucleus, endoplasmic reticulum, and Golgi apparatus. Cell body, which could be the cell body of the multipolar or pseudo-unipolar neuron, which carries electrical current away from the cell body. An axon if it's a multipolar neuron, but what would we call it if it's a pseudo-unipolar neuron? Central process. So axon in a multipolar neuron is a central process in a pseudo-unipolar neuron, which stores and releases neurotransmitter vesicles. That's going to be the synaptic end plate, also called the Bouton synaptic terminal. There's about 12 other names for it.
Those 12 will be on the exam.
That one I don't know. Maybe the end plate or terminal. Generally, I'll go with end plate or terminal. But you may see it as a Bouton, but you'll usually see the word synaptic in front of it, and that's kind of a clue. Synaptic terminal, synaptic end plate, Bouton, that's usually you'll see the word synaptic in front of it. Because there's so much interchangeable, you need to be able to recognize all of them. And we'll use it a lot more, even heading into the next chapter, which carries electrical current towards the cell body. Well, if it's a multipolar neuron, it will be a dendrite. If it is a pseudo-unipolar neuron, it will be a peripheral process, which transduces specific stimuli. This would be I should say peripheral receptor on this to make it more accurate, because we also have other receptors. So this should say the word peripheral receptor, and that would be on a pseudo-unipolar neuron. So I should say the word peripheral on here, and because we also have ligand gated and electrical, we have other receptors as well. So it carries electrical current towards the cell body of a multipolar neuron, which are characteristics of chemical synapses we did not cover. So I did take this one off, like synaptic delay, synaptic fatigue, we didn't cover. We started to cover the events. So what do you need to know about the events? What's the end result of an action potential? It triggers the opening of what channels? Calcium channels, and that causes the calcium to come into the cell, which causes the exocytosis of the synaptic vesicles. So that's what you need to know as far as we got into chemical synapses. That's what you need to know. We're going to cover chemical synapses when we get to the next chapter with the neuromuscular junction. So on this one, it is, they are limited by synaptic delay and fatigue. They do determine the direction that information travels because they are one way. Used between neurons throughout the nervous system, and they do use neurotransmitters for intracellular communication. But again, for our purposes, you need to know that it's the influx of calcium that triggers the exocytosis of synaptic vesicles. This is one of those where action potential triggers the opening of voltage-gated calcium channels, the influx of calcium, which triggers the exocytosis of synaptic vesicles. That's as far as we've gotten into the chemical synapse story so far. And so also we don't have gap junctions, but I did put 2 questions on there for sympathetic and parasympathetic. So we're going to draw lines on this one. Purple is parasympathetic. Red or pink is sympathetic. All right, what innervates sweat glands? Sympathetic, and that's the only glands innervated by the sympathetic system. What innervates lacrimal glands? What innervates salivary glands? What innervates reproductive glands? What innervates the SA node of the heart? Both. Parasympathetics will trigger it to decrease. Sympathetics will trigger it to increase.
What's the SA?
Sinoatrial, that's the pacemaker of the heart. So that's going to be heart rate. What innervates cardiac muscle? Mostly sympathetics, a little bit on the parasympathetic as well. Not a lot, but a little bit on the parasympathetic as well to decrease cardiac function. So we're going to say decreases heart rate and decreases force of contraction, essentially cardiac function. GI peristalsis. Parasympathetic is going to be the one that increases GI peristalsis. Iris dilator muscle. They're also called the radial fibers because they actually pull the pupil open like this, like a kind of a lines. So the iris dilator muscle is sympathetic. The circular fibers or the iris constrictor or pupil sphincters are going to close the pupillary aperture. They will be innervated by the parasympathetics, as will the ciliary muscles for accommodation of the lens so that you can read closer. Although as you get older, it gets a little bit presbyopia. Hey, Mom, you want me to hold that over here for you? Can you see it now? I had trouble reading my choir music now. Like, I can't see the choir music and the choir director at the same time. I get one or the other. Blood flow to the GI tract. It is a sympathetic function, but what happens to blood flow to the GI tract when the sympathetics fire? It decreases, vasoconstricts. So blood flow to the GI tract will vasoconstrict. Blood flow to the skeletal muscle and cardiac muscle will dilate. Those are both sympathetic functions, but they're opposite functions of each other. So we will increase blood flow. How about bronchi? I forgot to put bronchi on here. Smooth muscle of the bronchi. Innervated by both. Parasympathetic will be for broncho constriction. Sympathetic will be for broncho dilation. How about contraction of skeletal muscle? We said it could fit in box A, box B, or neither. This is the neither. Neither. This is neither sympathetic nor parasympathetic because it is not part of the involuntary system. It's part of the voluntary system. So that one's not innervated by either. Interesting. Questions on introduction to the nervous system. Again, you're getting 2 questions on parasympathetic, sympathetic. You're not getting questions on synapses or neurotransmitters yet, with the exception of the exocytosis of the synaptic vesicle. And you are getting questions on CNS versus PNS. Question?
For the bronchi, it increases parasympathetic.
How about I type it for you? Yeah, no problem. Parasympathetic will bronchoconstrict. And sympathetics will bronchodilate. Sympathetics use epinephrine and norepinephrine. So if you're having an anaphylactic response and your bronchi are going into bronchospasm and you can't breathe, you're getting a shot of epinephrine because it's going to trigger this pathway. It's going to trigger the bronchodilation. Good. I wrote a special question for this. These 2 questions I added in. So these are what I swapped out the chemical synapses and neurotransmitter questions for. Was your ANS questions. All right, chapter 4, transport of things across cell membranes. We're looking at 10 to 12 questions on this. This was a pretty heavy one, as well as 10 to 12 questions on chapter 5. So how does each typically pass through a cell membrane down its concentration gradient? Water, how does it go through a cell membrane? Typically through a pore. Sometimes it's called an aquaporin. aqua being water. You could figure that out, right? How do ions typically go down their concentration gradient? Typically through a transmembrane protein channel that may be opened or closed. What goes directly through the phospholipid bilayer? Gases, alcohol, and steroids. What binds to a transmembrane protein channel? And what do we call that type of transport? Because you may see it like this as the description, or you may see it as its name. Facilitated diffusion. And of course, we know if it binds that it is what type of transport? Broad category carrier mediated, which means it has to follow the rules of carrier mediated transport. So binds to a transmembrane protein channel, that is called facilitated diffusion because it is still down its concentration gradient. Did you need that again? Did you get that one? You good? Okay. Which will decrease the speed of passive transport through a membrane. So there was a slide that said, okay, these are things that increase the speed of diffusion through, and they include things like increase in temperature. What happens when you increase temperature? It increases the speed that molecules move and bump into, so it essentially increases the kinetic energy in the system. So if you increase temperature, you increase the speed of transport through that membrane. If there is a higher concentration difference between one side and the other, and you open a channel, it will have a higher speed of diffusion. Even on gases, if you have a higher concentration, this is how oxygen goes from being in your lungs to being in your bloodstream, because there's a higher concentration of oxygen in the air you inhaled than in the blood, and there's a concentration that pulls it across very quickly. Increasing the thickness of the membrane. That will decrease the permeability. It will decrease the permeability of the membrane for that particular molecule. It would be the same thing as if you were an ion or glucose, decreasing the number of transport molecules available or decreasing the number of channels. So that will decrease the speed of passive transport. If you increase the pressure on the gas or the pressure on the system, it'll have the same effect as increasing the temperature. And if you increase the number of channels or pores for that molecule, it will increase. Now notice how I could flip any of these and turn this into the correct answer. Everybody see how I can flip any of these? This is not a memorization question. This is a comprehension question. And a lot of it is common sense. If it's a thicker wall, it's going to be harder to get through. So if I decrease the thickness of the membrane, if I have a lower concentration difference, if I decrease the pressure or the temperature, or if I decrease the number of channels or pores, I can switch any of these and make them the correct answer. Does everybody see how that works? This is the kind of rewording that happens. Also encourage you to use arrows up and arrows down when you're reading the test question, because our brains start to get a little bit tangled up because I have a decrease in the question and I've got increases in here. Or I've got a decrease here and a decrease there. So I encourage you to use arrows up and arrows down on your sheet there to figure out the correct answer. Which is a transmembrane protein that may be opened or closed by a change in pressure. Mechanoreceptor, mechanically gated ion channel, also called a pressure gated. So they're kind of AKAs. When you see pressure gated, it's a mechanoreceptor. Aquaporin is for what? Water, and it's always open. What opens a voltage gated ion channel? Change in membrane polarity. What opens a ligand gated channel? Binding of something to it, like a neurotransmitter or a hormone, which is an example of a mechanically gated, of where a mechanically gated ion channel might be used. Okay, if you have a nicotinic receptor for acetylcholine or a chemoreceptor, what type of channel is that? That one's ligand gated. If you have a muscle spindle or baroreceptor, that's a mechanically gated. That's a pressure receptor. That would be a good answer to choose. Calcium channels for the sarcoplasmic reticulum. What is that? That's voltage gated. Sodium and potassium channels of the neuron action potential. Those are also voltage gated. Sodium potassium pump. That's not even a channel. That's another machinery. Did you go to that one? Again, name them as you go. Which is located at the axon hillock. Well, what happens at the axon hillock? Summation to threshold, generation of the action potential. What do you need to generate the action potential? Voltage gated sodium channels. That's what's going to start it, get it to threshold, which is usually found on the dendrites. Ligand gated sodium channels or other channels. Sometimes they're chloride, sometimes they're potassium, which is found at the axon terminal. That's another word for the axon, what? Synaptic end plate, Bhutan. So the axon terminal, what do we have there? That's where we have voltage gated calcium channels for the influx of calcium, which is often used to transduce environmental stimuli. Mechanoreceptors, mechanically gated. Not always, but often. Good on that one? Again, that's one question. I think I checked a bunch of that. I had like 3 questions on this last term. So I think I'm down to like 1 question on this one. So if you're still confused on this one, it won't kill you yet. I'll kind of spread it over a few tests. Carrier-mediated transport. What does carrier-mediated transport mean? Okay, first tell me the rules for carrier-mediated transport. Because if I swapped out carrier-mediated transport for its description, what would that look like? Which is limited by, what are the 3 limitations for carrier-mediated transport? And what's stereospecificity? D versus L. What else limits carrier-mediated transport? Competition. And saturation. What's another word for saturation? Transport max. What is transport max or saturation? It occurs when everything is bound and working as fast as it can. You're going to speed and then you're going to maintain your cruising speed. You can't go any faster than 80 miles per hour or whatever as you're going through. So another way to say this is which is limited by stereospecificity, or which is limited by competition, or which is limited by saturation or transport maximum. By the way, there's 2 questions on carrier-mediated transport. One of them is an example of what's limited, what's not. So is primary active transport? Yes, that requires binding. What's the keyword we're looking for primary active transport? How do you know it's primary active transport? It's going to use ATP and you're looking for the word pump. Facilitated diffusion. Who do we use as our example for facilitated diffusion? Glucose into cells. Requires binding. So it's the only one in this category that's down its concentration gradient. Countertransport or antiport and cotransport or synport are going to require the binding. What does not require binding? Simple diffusion. So any example of simple diffusion. Or I may just say simple diffusion. Questions on that one? Good? All are features or limitations of carrier-mediated transport except. So again, make sure you know these rules and the definitions of them. Binding sites of membrane proteins have stereospecificity. Speed of transport depends on concentration gradient. No, because that's not for carrier mediated, that's for simple diffusion. That's the rule for simple diffusion, right? Not for carrier mediated. Similar molecules can compete for binding sites. Membrane proteins have saturation points that limit the speed of transport. So this is a true. Speed of transport depends on concentration gradient. That's a true statement. but it doesn't apply to carrier-mediated transport. It applies to simple diffusion. Does that make sense? That's your oxygen, carbon dioxide, sodium. That's what drives your simple diffusion. Which mechanism pumps a molecule against its concentration gradient using ATP? Okay, glucose uptake into a cell using a GLUT protein. What is that? Facilitated diffusion. It doesn't have the word pump in it. Calcium pumped into the sarcoplasmic reticulum. This is a song in the old days. Well, who did pump up the jam, up the jam? Who did that one? You don't know. It was way before your time. But now you're going to, who's heard the song? There you go. Okay, well, at least that's good. Once I like give a quote for a movie that's my favorite movie and nobody gets it, I'm going to be like, okay, I'm out. I'm retiring. Here's my resignation letter. Although my spring terms are still the ones that keep me young because you tend to have more diverse age populations and other things coming in. Chloride exchange for bicarbonate. What is that? What's that an example of? Countertransport or antiport. Concentration of iodine in the thyroid cells. That's going to be actually a synporter. It doesn't have the word pump in it, so we're going to assume it's not. Most of the time you're going to have pump in it. Concentration of iodine, this is cotransport. What's the energy source for primary active transport? ATP. What's the energy source for secondary active transport? Somebody else's concentration gradient. So chloride with our exchange, or sodium for our exchangers. What is the energy for passive diffusion, passive transport, or diffusion? It's another way to say concentration gradient. What type of energy is it? Kinetic energy. So passive transport or diffusion is concentration gradient or kinetic energy. Primary active transport is ATP. Secondary active transport is the concentration gradient of another molecule. So that one's a little bit, which is true of ion movement through the ATPase pump. Okay, who goes what direction? When I get this question, I'm going to write that not beforehand. When I get to this question is when I'm not going to read the answer choices until I type out 3 sodium out, 2 potassium in. So before I try to answer this question, I'm going to write this on my paper. Because otherwise what happens when I go look at my answer choices is I start to get myself tangled up. Now I'm not going to get myself tangled up. I'm going to look at the one that goes along with what I know. Okay, 2 molecules of sodium enter the cell. No. 2 molecules of potassium are pumped into the cell. There you are. 2 molecules of calcium. Nope. 2 molecules of chloride. Nope. 3 molecules in, out. 3, what direction? You're getting one of these questions. What direction? What ion? When sodium level increase, the blood becomes hypertonic. If it's not corrected by ADH, what happens in the body? Cells placed in a hypertonic solution will shrink. Because water wants to go to where there's more particles. Again, draw it out if you have to. If there's more water, if there's more particles out here than inside the cells will shrink. There are 3 questions on carbon dioxide and water on the test. Because we did draw it out. We covered it. There are 3 questions on carbon dioxide and water. Again, because I took out the neurotransmitter questions. So make sure you know this one. Oh, no, like there's more than 3. So there's 3 questions on this content. I took out the neurotransmitters and the other ones, and so that's where I subbed in the ANS, PNS questions, and I gave you 1 extra question on here. There used to only be 2 questions on this one. So do we need to draw this out? I think maybe we need to draw this out, right? What is this question? Or everybody comfortable with this one? Let's answer the questions because we don't have time. What combines with water? What combines water with carbon dioxide to create carbonic acid in the red blood cell? Carbonic anhydrase. What transport bicarbonate out of the red blood cell? Chloride exchange. Remember, bicarbonate is HCO3 negative. So we need to change a negative for a negative. The chloride wants to come in because there's not very much chloride in there. The bicarbonate does not want to go out. So the chloride coming in expels the bicarbonate. What buffers the hydrogen ions in the red blood cell? Hemoglobin. How does carbon dioxide move through a cell membrane or a capillary membrane? Simple diffusion. Which is false concerning movement of water across the cell membrane. Water usually moves easily between intracellular fluid and extracellular fluid through pores. True. Water moves into the compartment with more osmotically active particles. True. Water moves into the compartment with higher hydrostatic pressure. Hydrostatic pressure pushes water. Oncotic pressure pulls water. So it will move into the compartment with lower hydrostatic pressure. Body water equilibrates across capillary membranes fairly easily. How is sodium typically absorbed into an epithelial cell from the GI tract? Okay, so what? We didn't cover this. We did. Let's look at it. Okay, this is the cell. This is the GI tract. First thing you need to know is sodium an intracellular ion or an extracellular ion? It is extracellular, so it means I have more sodium in the GI tract after eating that bag of Doritos than I have inside the cell. So how are we going to get sodium from the GI tract into the epithelial cell? If there's more what the bleep? Come on now, behave. We're done. If there's more sodium, how does it, does it need active transport or can it use passive transport? It can use passive transport. It's going to go down its concentration gradient. God bless you. If we were going to get it out of the cell and into the bloodstream, that would require primary active transport. What uses pinocytosis? Large molecules. So sodium's going to be absorbed from using passive transport. All you have to know on that one is intracellular or extracellular. It doesn't matter where you put it. If you know sodium is lower concentration inside the cell, you know passive transport will get it in. You'll need active transport to get it out. Does that make sense? How do larger molecules like protein typically enter a cell? Pinocytosis or receptor-mediated endocytosis would be a more accurate one because these are all too small for a large protein to get through. All right, 12 to 13 questions on membrane potentials. Which cells are excitable or have the ability to use a local change in membrane potential to trigger an influx of calcium ions? Macrophages, yes. Glandular cells, yes. Ciliated or flagellated cells, yes. Skeletal muscle cells, yes. Neurons, yes. All of the above, yes. Although I don't give you all of the above or none of the above questions because boards doesn't either. So boards doesn't give you that type of question, so I don't either.
Examples that go on.
Epithelial cells. Make up something that's not one of these and throw it in there. Hepatocytes. Right? What else? Anything that's not one of these. Red blood cells. Right? So anything that's not one of these. Red blood cells, hepatocytes, epithelial cells, skin cell. I don't know. Throw something in there. That's not one of these structures. Good? It took me a little while to come up with some examples as well. So those are the ones that are on the test. All of the following affect the Goldman equation except what's the Goldman equation? Nernst potentials for every ion. So if it affects the Nernst potential, it affects the Goldman's equation. How easy is it for that ion to cross, and what will determine the membrane? So the polarity of the electrical charge of each ion, yes, that will affect the Goldman equation because it affects the Nernst potential. The concentration gradient of each ion, yes. Another way to say that would be the electrochemical gradient, right? The permeability of the membrane to each ion, the temperature difference of each ion. What the bleep? Can you have a temperature difference of one ion versus another ion? No, they're in the same environment. Temperature is determined by the environment, so that doesn't even make sense. Which ion is attracted into the cell by its charge but repelled out by its concentration? So the question is, it's attracted in by its charge because it must be positive, but it's repelled out by its concentration gradient. So I'm looking for something positive that's higher inside the cell. What are my two options? Potassium. What's my other option that's not on this list? Intracellular ions. What do you got inside the cell? Magnesium and the P's. What's the charge on magnesium? I could sub potassium for magnesium. That would also work here. I could use chloride and word it for chloride or sodium or calcium. So you have to know the charge and the concentration gradient for all sodium, potassium, chloride, magnesium, and calcium. How many molecules of sodium are pumped by the sodium? What's the primary function of the sodium potassium pump? Create the concentration differential for both sodium and potassium. What triggers the contraction of the cytoskeleton? Influx of calcium. What creates most of the negative charge inside the cell? Leak of potassium or efflux of potassium. What rapidly changes the membrane polarity? Voltage-gated ion channels. You do know a lot. Look at how much you've learned. All are characteristics of neuronal action potentials except, do they have variable sizes and shapes? Stereotypical size and shape for a given tissue. Self-propagating, non-decremental. Depolarization occurs when threshold is reached and not before. Variable speeds of conduction, larger and more myelinated is faster. Temporal summation. Stimulatory inputs in rapid succession. Spatial summation would be simultaneous. This one would be spatial summation. Which channels are open during the depolarization phase of a neuronal action potential? Voltage-gated sodium channels. Which are open during the repolarization phase? Potassium channels. When is the neuron membrane least permeable to potassium? Remember, at rest, you've got potassium leak, and then during repolarization, you've got potassium going out. So it would be during depolarization where you have the least amount of potassium crossing the membrane. Absolute refractory period occurs during all of the following except. All right, make sure we read absolute and not relative. All right, so I'm looking at this box here. All right, depolarization, yep. Overshoot, yep. Repolarization, yep. Undershoot, nope. What does undershoot correspond to? Hyperpolarization after potential effect or relative refractory period. All right, I just wanted to finish so you had all the answers. What increases conduction velocity for a neuron? Increasing the width of the axon and increasing the myelination. Okay, and then we're not doing synapses yet. Motor units, the smaller the muscle, the more fine motor. There is one question on motor units, as it was at the end and we did mention it. Large motor units are where you have gross motor control. Small motor units are where you have fine motor control. So which muscle is going to have the largest motor units? Pick the biggest muscle. In this case, it would be deltoid. But I have small muscles and I'll have one big muscle. So pick the big muscle. Cool. Thanks for coming today. Sorry for going over, but I think it was worth it to get through the entire review. I will see you tomorrow. Please sit in your assigned seats. I will bring you paper if you want to use it. Don't take Dr. Shaw's big sheets. I'll bring you your half sheets. Any other questions? I'll be in my office hours today, 11 probably till 12:30. Yay. That I'm not sure, Reese, because sometimes I've got on the quiz, I've got some questions that I took off of this exam, but they might be on the next exam. Correct. No questions on chemical versus electrical or neurotransmitters, but there are CNS versus PNS, and then the one with calcium you need to know. Yep. As far as I know, no. But I can't guarantee that 100% because if it's on the study guide, most of what I got on the exam, I did get on this review, but I can't guarantee it's 100%. You may see one question that's on the study guide that maybe didn't make it to this review, but they should be pretty close.
Correct answer possibly. Let's say like Toxigio, 1, 2, 3. It would be 1 versus 3 in that sense. That's the question. It's always going to be 1, 2, 3 for Toxigio.
Generally, you're going to say 1 for Toxigio because that's the board's answer. But I will either give you 1 to 3. Like it'll be obvious. I don't. Yeah, there shouldn't be 2 correct answers.
Okay.
I'm very careful to not put 2 correct answers on a test. To the point where even in Phys 3 now, somebody came up and was like, oh, I read this question this way. And I was like, I never occurred to me that you could read that question that way. And I see now that you chose a correct answer. So I will give you credit for that.
Okay.
But yeah, I'm very careful about making sure there's not 2 correct answers.
Okay. Thank you very much.
Yep. All right. See you tomorrow.